So Rosie, would you say, are you a geneticist?
I mean, in terms of my research program, I'm sort of a bacterial evolutionary geneticist. In terms of as an educator, I've most enjoyed teaching genetics. I've taught genetics a lot. All the lectures that were created for the massive online open course Useful Genetics are on YouTube now. Just Google Useful Genetics and you'll find this YouTube channel with about 150 10-minute videos explaining genetics. And they're still pretty damn good.
Oh, they're very good. I watched them all last year. They're still very good.
And they're in many ways a lot better than what you get in a genetics textbook because I've thrown out much of the crap that we still teach, that we still focus on in conventional genetics courses. Mendelian genetics is still in there, this simple one gene, one clear distinction. It's still in there, but it's downgraded to just one module rather than taken over, especially since David Suzuki's groundbreaking genetic analysis textbook. The emphasis in genetics textbooks has been teaching people how to be scientists who use genetics as a tool to understand how inheritance works and how our bodies, organisms work. So it was very much doing crosses between organisms with clear genetic differences and looking at the offspring and looking at the same properties and saying, what can we learn about how the gene works, how the gene is passed on by looking at the results of genetic crosses. Lots of work with fruit flies, lots of work with mice, lots of work with corn. And these were very good courses for training people to be geneticists. And there was this sort of uniform, well, perhaps not entirely uniform, but widespread sense in biology that the geneticists were the smartest ones. Genetics was the hardest. It trained you to think rigorously, and geneticists were very smart.
They are very smart.
A lot like physicists. But that's not what we should be teaching our students. Almost nobody is going to go on and be a classical geneticist anymore. Some of our students, maybe a few, will go on and become modern geneticists, doing a lot of stuff with DNA, real fancy high-tech sort of stuff that doesn't really involve crosses at all or Mendelian genetics. But we're still teaching our students Mendelian genetics as if that was what really mattered. And that I thought was a terrible injustice to the students. Students find genetics very hard. They work very hard to understand it, and it just seemed really unfair that we are asking them to do all this work that isn't going to benefit them anymore. They will never have a use for this. The odds are very small that they will never have use for this.
There probably weren't a lot of female tenured professors at UBC when you got that, right?
When I first came to UBC, I joined the biochemistry department, and there were about 15 other faculty members, one of whom was a woman, who was quite disgruntled with her life and really only lived to play golf for various reasons. It was a soft money position, so my job security was not great. So I wound up taking a hard money, secure position in the zoology department, which shortly before they hired me had about 40 positions, of which 1/3 of one position was a woman. Which is to say, there was one woman who had a 1/3 time appointment in zoology and a 2/3 time appointment in agriculture. At the time I actually joined the department, they had hired another woman a year or two before, and then they hired me and another woman, and then they just kept on hiring women. Probably as many women as men in the zoology department. And there were lots of women in the biochemistry department too, but at that time, no.
So there's all the other professors, and you go down the hallway, and mostly very serious and not as approachable as Rosie. And Rosie would have this fiery red hair one day, and then fiery blue hair the next day. And so maybe tell everyone a bit about your educational background.
My educational background. So I started in the late 60s, went to Catholic school, after grade 10, managed to convince my parents to switch to public
So
school.
where were you born?
Born in North Vancouver, actually. Grew up in North Vancouver. Started at university at SFU thinking I was going to be an engineer. Engineers looked like more fun than anybody else. I was good at science, and engineers definitely looked like they had more fun. But I was at SFU to teach engineers, so I was going to do first year science there and switch. But it was the 60s, and so instead I turned on that TV and dropped out.
Okay, yeah.
All the sort of creative, intelligent people were doing that. The nerds were still doing nerdy stuff, but the rest of us were.
Okay, so what did you do there? You dropped out.
I dropped out. I moved out of my parents' house to live in a student commune in New Westminster, which was getting its act together to become a student cooperative and actually buy some property.
That's cool.
I wasn't one of the leaders in this, but I was part of the group. We got some seed money from the credit union or something in Vancouver and used that to get a mortgage from Central Canada Mortgage and Housing, whatever, CMHC, which had a special program for student housing. And so we bought 2 big old houses in New Westminster side by side on what is now very valuable.
Yeah.
And renovated them to be inexpensive student housing that was getting run as a co-op. We paid $75 a month, room and board. Standards were low. You might not get a private room, board. There was plenty of food. Rice and
I mean, that's such a great thing. You can't even do that.
That co-op is still going.
Wow.
Well, at least it was a few years ago. The 2 houses are still standing and still
So
in New Westminster.
you were one of the originals in there?
Yeah, still occupied as a co-op, not really much of a student co-op anymore. Still runs as a co-op.
That's cool. They should have more of those.
Yeah. Anyway, so I dropped out after. By the time we were becoming a co-op, I'd already dropped out.
And you did a hippie thing? Like,
Did sort of hippie things. And then a few years later, I had a baby, got married, more or less same time. Decided to go back to school. Decided I wanted to go to medical school. Went back to SFU, did the rest of first year science and second year science. And then we moved to Australia because my husband had just gotten his PhD in math and he'd gotten a postdoctoral position in Melbourne, Australia. And I would never have had the imagination to leave Vancouver. I was a typical, why would I go anywhere else? It's nice here, sort of Vancouverite. And Australia was just such an eye-opener in terms of, it's so different in ways that I never thought things could be different.
what do you mean?
The light switches work the other way around.
Okay, yeah, yeah.
It's like, what? Things you thought of, that's a perfect example, because it is something that you had thought of as sort of just natural, that up is on and down is off. But in Australia, up is off and down is on.
Okay, yeah.
And you realize, oh my God, I've been taking for granted all this stuff that is not necessarily true. Anyway, so I did an undergrad degree in Australia. First thinking I was gonna go with my husband and go to medical school, and then decided, no, I'm gonna do molecular biology, because molecular biology was really cool.
Yeah, what year is
This
this?
was like 1973.
Okay, so that helps you get into the molecular structure of DNA.
Yeah.
That stuff.
Yeah, I
Yes.
did a honors thesis on mitochondrial DNA in yeast.
Okay.
So I was running gels and staining DNA and things like that.
Yeah, that was cool at that time.
In
It was
the
kind of all new, right?
Mid-70s, yeah.
Is it circular as well?
Yeah, it's circular.
Double-stranded but circular.
So everybody's mitochondrial DNA is descended from bacteria that are the ancestors of mitochondria. And it's circular.
Okay, just for me to understand that a bit more, the mitochondria was probably living on its own at some point.
It was.
Long time ago.
It was free living bacteria that were engulfed by bigger cells. And rather than becoming digested, they were sort of co-opted to become part of the cell's machinery for generating energy. Because the bacteria were really good at generating energy.
And that must have happened millions of times before birth, right? Billions of times.
Big cells have been eating bacterial cells billions and trillions and gazillions of times. And some of the time, almost all the time, the bacterial cells are just food and they just get digested.
Yeah.
And sometimes they stick around for a while. They're useful.
But it only
And
happened twice, right?
Well, there are lots of examples where the cells stick around for a while.
I thought it was only, I guess nucleus, chloroplast, and mitochondria.
But then, if you look at, say, single-celled organisms that live in plant water, they're eating bacteria all the time. Sometimes the bacteria just stick around for a day or a week and get passed on to multiply inside the cell, live happily inside the cell. Amoebas are a lot like engulf bacterial cells, but they don't necessarily kill them. The bacterial cells have found ways to exploit the living environment. Chloroplasts take sunlight and water and carbon dioxide and turn that into energy. So those are the two times when it worked really well. We know that those cells, those structures used to be bacteria because they still keep part of their bacterial chromosome.
Oh.
And that's the mitochondrial DNA or the chloroplast DNA.
Oh, yeah, that's interesting.
It's extremely cool.
Yeah.
And you can tell when you sequence the DNA, say, oh, these genes are a lot more like bacterial genes than they are like other genes.
I see. So it's not just a wild guess that that's how it
Oh
happened.
no, we know.
You can actually track sequences going back to similar.
Yes, absolutely.
Yeah.
We know that's very similar. There's tons and tons of evidence.
And then between the two, like as a planet, if you look at living things on the planet, would you say the chloroplasts are more important than the mitochondria? Can you say something like
If
that?
it wasn't for the chloroplast, there would be no food for the
Yeah.
mitochondria to work on. 99.999% of the energy that is being used by organisms on this planet was made by photosynthesis. It was made by chloroplasts or by photosynthetic bacteria, like the ancestors of chloroplasts.
Yeah.
They take
Either a tree or like some kind of algae or something. Right?
pond scum,
Yeah.
everything that's green, and even a lot of things that aren't really green, are gathering energy from the sun and turning it into what the rest of us treat as food.
Yeah, so it can link things into like hydrocarbons.
Take the carbon dioxide and water atoms and rearrange them to become sugar molecules.
Okay, and that's carb.
And that's what photosynthesis uses the energy from the sun to take the hydrogen and oxygen that's in water, carbon and oxygen that's in carbon dioxide, and rearrange them and stick them together into more complex forms. And the usual form is a simple sugar. And then that simple sugar can be converted by other enzymes into all of the building blocks of our bodies.
Do you think that's cool?
Oh my God, so amazingly cool.
Yeah, it's crazy.
It's astonishing.
When were you an undergrad?
I was an undergrad in the late 60s, early 70s.
So you're thinking about this stuff for like
Yeah.
60 years or
50
something.
Years, yeah, absolutely. Well, there's this worry that undergraduates often express. What if the scientists have already discovered everything? What am I going to do? There'll be nothing left for us to learn. But it doesn't work like that because we have still just scratched the surface in terms of, we say, oh, a breakthrough, now we understand this. But we're still just scratching the surface, and there's a ton of complexity that we don't understand at all yet. That we're not going to run out of things we don't understand. So anybody who wants to be a scientist, there's plenty of opportunity to find out amazing new things.
Of all the undergrads, students you've met, how can you tell someone's meant for science? Like when I was with you, I was very eager and did the experiments and tried hard, but I kind of knew after several months, I'm like, I don't know, Rosie's just,
Yeah.
you know, at a different level and at a different level of enthusiasm.
Kind of motivation. I remember
Yes.
Naomi Fast.
Yeah.
Naomi came to me as an undergrad. She came to my office and
Okay.
said she wanted to do research. And she explained that she'd been reading the biography of Barbara McClintock called A
Okay,
Feeling
yeah.
for the Organism. And she had read the part where McClintock, for some reason, she's not allowed to get her research field. There's a fence or the gate's locked or something. And she's climbing over the fence to get in
Okay.
because she wants to find out what's happening with the corn. And Naomi was saying she wanted to be that person. She wanted to be doing work that was that compelling that she would climb over the fence to
Gee.
get to. And I thought, all right, this one's a keeper.
So is it,
but
is
it's-
it intellect? Is it level of enthusiasm? Because people can feign kind of
I've
enthusiasm.
had enthusiasm without critical thinking, and that's not been very successful. Like, it's easy to have ideas. The hard part is wanting to know if your ideas are right or not, and being able to not, as opposed to wanting to show that your ideas are right.
Okay, yeah.
Versus wanting to find out whether or not your ideas are right.
I see.
These are the people that are going to be good scientists. The people who just want to show that their ideas are right, or who are just producing ideas but not thinking about how to test them.
So is it a seeking of the truth? Is it different?
It is the wanting to find out what's true as opposed to what you would like to be true. It's hard to assess that
It's hard to assess.
in people.
Well, even the scientists, some of them, it's just like, they
just
got
know
lucky.
They're right, yeah.
Or no, like some of them, like, wasn't penicillin just like a lucky find?
Actually, no, you know.
I thought he was a dirty guy that left it over a weekend or
Oh,
something.
well, he was, so this is Alexander Fleming. He was working in London at a hospital, and he had a little sort of microbiology research lab, and I actually went on a tour of his little lab, which is about, kind of this big total.
Wow.
It was just two little rooms. And he was studying, I don't know, fungi and bacteria.
Yeah.
But he had discovered the enzyme lysozyme.
Okay.
I think from snot dripping from his nose when he had a cold. He was a noticer. So he had a plate with
yeah.
bacteria growing on it, and he noticed that where this drop of snot had fallen, that the bacteria were all getting dissolved.
Huh.
And he purified this and showed that there was an enzyme that actually broke down the cell walls of bacteria. So they died and nursed. And this is an enzyme that we use, microbiologists, molecular biologists use this all the time to like break open the cells of E. coli. You add lysozyme, everybody's got to do a lysozyme in the freezer. So he had discovered lysozyme. And then he was independently working on a lot of other stuff. And he had bacteria growing on a plate.
Yeah.
And he went away. He came back after, or he set it aside and
Yeah,
forgot about
it was
it
a
for
while, yeah.
And then he looked at it, and he said, So this is what he did. He was the kind of person who would look at something and say, that's peculiar. So that's a really important.
So curiosity, or no, observation or curiosity?
Noticing when something is.
Okay,
So it's noticing when something isn't what you would expect.
I see.
That's really important in science, because you do an experiment, you go, no, there's the controls, they all look fine, and then you take it to your supervisor and they go, what about this thing over here? What's this blob over here? What's this little squiggle down here? And you go, do they matter? Why is this blob bigger than this blob? And it turns out that these are important things. So he was a person who would notice. And so he noticed that around where the fungus had grown, the bacteria couldn't grow. And partly because he had already studied lysozyme, which killed bacteria by spreading through the agar and preventing them from growing by bursting their cell walls. When he saw that something from the fungus was having the same kind of effect, he was prepared to notice that this particular thing was important. because of, so partly he was a noticer, partly he had done this prior work, and he made the connection that what he was seeing here was like what he had been seeing in License. And so it wasn't just that he involved. He was prepared by his prior work, and he was attentive to noticing whenever things were going on that we didn't expect.
So being a good noticer might be a common trait among scientists?
I think so. Looking for anomalies, looking for that doesn't make sense, and it's at all levels of. I mean, it can be in a classroom and somebody says
Could be
something.
math.
It could be math. It could be chemistry. Go, wait a minute. But I thought this did this. How can this do that?
Yeah.
That kind of, wait a minute, it doesn't make sense. And having the confidence to not just say, oh, I must just be stupid, I just made a mistake. But instead, pushing at that doesn't make sense. Or there is either the not having enough confidence, or there's a related thing of, I don't even want to ask about stuff I don't understand, because I'm just gonna feel more and more stupid.
That's a big one. That's an interesting one you bring that up, because I remember in some of the whiteboard talks that you did with Andrew, Rosie is asking such simple elementary level questions that you wouldn't let us go on to a different aspect of it until we understood a certain core element, a fundamental component that seemed kind of easy, but we spend 15, 20 minutes on this concept.
A lot of students are afraid when they feel like maybe they don't understand something, they're afraid to probe into it because they're not confident that they know how to learn. Whereas I'd go, oh, I don't understand this, but I can figure it out. I can learn it. I know I can come to understand it. Whereas they are, it's like they sort of feel like they're predestined for failure. That their past experiences have left them with the sense that they're not going to succeed at learning. And so they won't even, when they think they might not understand something, they don't even want to go there. Just back away. I mean, it's like you have a bruise or something and you don't want to even touch it. It's like, I don't want to, because something bad will happen if I try. Oh, and it's crippling.
It's crippling because then you're stuck and now you don't understand the first principle.
And then
now they're on 3, 4, 5.
Yeah. And they won't ask. They will not ask questions.
Okay, but the tough thing in that situation, I can remember being back at that stage is, there's no dumb questions, but there kind of are some dumb questions, right? A little bit.
Oh, yeah.
Where it's just like, okay,
Some questions are certainly smarter than others. And the
so
guys in the
you don't
front
want to
row,
be, you don't want to be the guy asking the dumbest question.
wait, what's an atom again?
Yeah, well, you can't do that, but then how can you pick your spot and be like, well, just wait, this is important, and then the other smart people are like, okay, that's important.
So if you notice the guy next to you, say, what's an atom? And if he goes, see, this is what I did as an undergrad, was I sat next to the smartest guy in the class, in biochemistry class.
Okay, that's a good idea.
And I never said, did you understand that? What's that mean? And I go, excuse me, sir, because it was sir. We didn't understand that. Could you explain it again?
That's a good idea. So
And-
having a partner in crime.
Well, knowing that we were the smartest kids in class helped a lot. Whereas, so in science one, I mean, there were really smart kids, and there were smart guys, especially in the front row, and asked intelligent questions. But they were the kind of show-off questions that you ask to kind of demonstrate that you understood what they said, and now you're asking them a more refined, complicated interpretation or something. Complicated. Well, what about this? And how does this relate to that? And the students in the back rows are going, oh, I have no idea. I can never ask questions at that level. And so they won't say anything. Whereas what we want is could you go back and explain that again? I didn't understand it. Those are the questions I really want to get from them. And in the end, I figured out a way to do that. But which was by letting them ask them anonymously.
So I tell them, keep track of what you didn't understand, write down the slide number. At the end of the class,
Oh, I see.
here's a, clicker thing. You can submit anonymously your questions. And then at the beginning of the next class, I'm gonna go through your questions and answer them. And so that worked. Students would say, oh, could you go over slide 18 again? I don't know what this word means. And I would take the first 10 or 15 minutes of the next class just going through the questions from the last class. And I felt like I griped so much at them about not asking questions that I felt like I had to honor all their questions by answering every question that they posed at the end of the class. And that worked, they did. Then they would
That's a good idea.
ask, but it's a shame to have to wait till the next class to get an explanation. But I can see how they are intimidated by the smart guys in the front row. Especially they got these big testosterone voices. Yeah. And they're little Asian girls with tiny little squeaky voices.
But that's what's so amazing about you too, is like, you're not a big person, right? But you have a big brain.
I'm the oldest of a big family.
And you don't, I don't know, big mouth is not a good comment to have about
Well,
someone.
I can talk very loud.
But Rosie can stick up for herself easily,
Yeah,
I would say. And you're not intimidated.
No.
And so how do you get that? Is it the knowledge first? You got some A's and you're like, I feel more confident now. I could go out and say, even when you were an SFU dropout, you were probably still the same person. You'd probably just speak your mind.
I remember in a chemistry class, I think this would have been first year chemistry, and we had a midterm, and we'd done bad. People had done badly, and the prof was up at the front, and he was giving us a hard time about a question that asked us what was anomalous about something, and I put my hand up and I said, perhaps part of the reason people had difficulty with this question was because you'd spelled anomalous wrong. Because you have again. What a pain in the butt I was. Because he had spelled anomalous wrong. And he spelled it wrong on the exam too. And he was giving us all a hard time for not knowing what anomalous meant. It is a learning feedback thing.
Because you need to, I would say as a scientist, you kind of need to be a little bit brave too, right?
Well, you have to not be afraid of being wrong. You should be comfortable with being wrong. Like not feeling like you have to pretend you were right. You say, oh wait, I don't understand. Is this how it works? Oh no, it's not. Oh, I see now.